Good afternoon, everyone. So it's my pleasure to be here in Bordeaux. I didn't know that Bordeaux was a tropical city. So now it's done. So we are talking about clinical trials in prevention of Alzheimer's disease. And I have to give my disclosure and to say that I was involved in large clinical trials, so I'm a little bit biased and favorable to the multi-domain preventive trial. So what we have learned so far in the field of prevention of Alzheimer's disease and dementia. So we did a few years ago a summary of all the published large or not large preventive trials that have a good methodology. And the surprise was to see that most of the trials were negative. We have a huge amount of paper that find with good methods some negative study, and less than 20% of these clinical trials, preventive trials, were positive in the field. And among this small amount of paper that was positive, we have one multi-domain intervention that was just published at that time. It was a finger trial that we will see in details. So why do we have all this negative trial, and why is it so difficult to demonstrate an effect? And we will see that during the presentation. So for most of the people, when a trial is negative, the conclusion is that this intervention have no effect. This is a regular comment that we have. We missed the point. But in fact, there is two other explanations. Another explanation is maybe there is no effect in that particular trial because we decide the dosage, we decide the critical window of exposure, we decide the duration of the intervention, and maybe is not the good way. We didn't choose the good way. Another point is that maybe there is an effect within the trial, but the methods that we use are not optimal to detect a small effect, because we need to remember that we are in prevention, so everybody is normal, nobody has the disease, and so the efficacy that we can expect could be very small. So we decide to move to the multi-domain prevention trial for different reasons. This is a surprise, this nice picture. You know that since yesterday, or even before, that there is an amount of protective factors against Alzheimer's disease. And I'm completely in line with the comment of Cecilia yesterday, who say that, of course, there is these 14 modifiable factors, but we have more and more evidence for other factors. And I think about nutrition, I think about sleep, and maybe more. So it's at the minimum these 14 factors that we can analyze in trial. Another thing from EPI study was a paper from Nick years ago that found a synergistic effect of two protective factors like physical activity and diet for people who, in New York, who have a good diet and good physical activity. It was they are less likely to develop Alzheimer's disease in comparison to the people who have got only one of these protective factors. So we will see here a sort of summary of the multi-domain preventive trial. So by doing that, we choose the randomized control trial that testing a multi-domain intervention, which is a combination of lifestyle-based approach and sometimes a dietary supplementation for the prevention of AD, incidence of dementia, or maybe cognitive decline, on even more on biomarkers that is a sign of the disease. And we select only the paper that exclude at baseline the demented people. It seems very frequent to do that, but some of the paper did not do that. So as it is a preventive trial, we select only this paper, and we select also the paper who have got a priori a sample size calculation based on the cognitive outcome. And for this particular presentation, I focus on a large-scale trial to avoid to have too many. So now we have 29 trials in the multi-domain area. 26,000 people were included in total in this multi-domain intervention trial. And we can separate this trial in two phases. The first phase were from scratch. In fact, we start all, I think, at the same time to recruit the participants. So we were three teams start exactly at the same time in 2008 in order to recruit the population. And after that, we have some people who include the recruited population in 2009, 2011. What was remarkable at that time is that we didn't have any publication in the field of multi-domain intervention to guide our trial. So when we are doing a trial, it's always good to have good data in order to base our hypothesis. So in order to base our hypothesis, we did that from the epidemiological study, and we know that the population included in trials are different. So we will not go within the details of all these 29. I focus my talk on few of them. So for each line, you have a green line when the primary outcome is positive. So I will start with the lead trial because it was published very early into 2015. And what was interesting in that trial, I think it was the first personalized preventive trial because she asked a health worker to define specific goals with the population in order to prevent the cognitive decline. And with a very simple test of the MMSE, she found a positive effect of this intervention. It was a multi-domain intervention, very classic, combining cognitive activity, physical activity, nutrition, alcohol reduction, tobacco cessation. That's what they ask. And what was interesting in that population is that she had 5 arms. The only thing that differed between arms was the intensity of the intervention. And in fact, the more intense intervention arm was the more effective. And within that arms, she had something that is not so frequent. She gives some reward for the people who were adherent to the intervention, something maybe to keep. So after that arrived the finger trial. The finger is really the landmark of the multi-domain intervention trial. It was published in 2015. So she included 1,200 subjects, followed for 2 years. And the people who were included in that trial came from a cohort. So they recruit only people who have a high risk to develop AD based on the CADE index. And if you don't know the CADE index, you have this, and you can just calculate your score. I did that yesterday night. I say, oh, wow, now it's changing, so I stopped to calculate it. So, but the results was positive for this particular trial. So just to give you an idea about the complexity of this type of trial, so you have a combination of group session, individual session, on nutrition, on physical activity, cognition. They have to have the visit of the nurse every 3 months during 2 years. So it's a very demanding trial. And what was interesting in the type of intervention is that in the finger trial, they start with a low level of physical activity, and during the trial, they increase more and more the intensity of the physical activity. But usually the trial starts hard, and then they have a decrease phase and the maintenance phase, like a step-down approach. And this is, I think, interesting in that study, this difference. Another study was interesting, published in the JAMA by Rovner in 2018, because they focus on people that need maybe some intervention, and in that population, maybe the intervention could be could have a huge benefit, and so is what they find in the Black individual who have already amnestic MCI, and so they have a positive trend in that store. And there is also few more that were positive, but nothing new. Then arrived a very bad period for us. All the trials were negative, even big one, even well-designed one. We have this dark, I would say dark phase. So in this presentation, the green bubbles are the positive trial and the gray bubbles are the negative trial. And the size of the bubble is dependent on the number of subjects included. So we were close to the people that designed the Prediva study in Netherlands, which was an intervention, multi-domain intervention based with a nurse approach that see very frequently the person. They have more than 3,000 people followed for 6 years, but the outcome was a dementia incidence, and they miss the point to demonstrate that. After that, we have this list of negative trial. And then arrive since 2024, so since 2 years, we have a nice paper, nice positive intervention that I want to focus. So this morning, Nick discussed about the SMART trial that was designed by Christine Yaffe. This trial is important because it's a personalized intervention. The health coach go to the person to visit and define some goals, and they include people who are at risk to develop AD, and they found the positive results on the cognitive outcome, but also on the composite risk factor score, which could be a surrogate or something near the cognition. And then we were part of the ProDemo study because, as you see for finger, these trials are very complex, and we wanted something very light. So with the ProDemo study, we moved the intervention with a smartphone. So there was a coach in behind that gave information, but all was done with a smartphone to decrease the burden of the study. And 1,400 people were followed for 1 year and a half, and we got a positive signs on the dementia risk score with a CADE score. Almost at the same time, there was another study in Germany who did already the same, but the result was negative. They chose another dementia risk score, the Libra score. So it's difficult to do a synthesis because the number of subjects are quite the same, the design are quite the same, except the modality to deliver the intervention. And then we have the US pointer. So usually in this kind of trial, we can have a placebo group, no contact group as a control group, or we can have an active group. In US pointer, there is no control group. She just compared, is the paper from Laura Baker. She compared two types of intensity of the same intervention. A very intense intervention in comparison to the self-guided intervention that could be very useful and very easy to implement in the general population. So she included people with a high level of risk, and after 2 years, in 2,000 people, she found some positive effect on cognition. And then arrived the Maintain Your Brain study, which is completely online. All is online. The intervention is online. The assessment on cognition is online. So Henry Brodati in Australia include 6,000 people followed for 3 years. And he found a positive effect on the cognitive score. At the same time, the Apple Tree do the same online trial with a very low intensity of the trial in order to be easy after that to disseminate in the population, but they miss the point of the primary endpoint. After that, we have some positive and negative. So what we can say about all these trials? So if we are looking at the type of intervention that trialists use, they use physical activity. Almost all the paper have a specific group or individual action on physical activity. Then after, the cognitive activity arrive just after, and nutritional counsel. And in the less important range, we have the management of cardiovascular risk factors and social activity. And for few of them, we have smoking cessation, alcohol reduction, sleep management, chronic disease management, and even depression management. So if you remember the nice figure of Jill Livingston, we now all this trial finally go through these risk factors, but now if we are doing just a summary, we have half positive and half negative. So it's difficult to do to do a real simple summary. So what are the challenges by doing that, and why we have positive and negative study? One of the challenges is to recruit the population that could benefit from the intervention. We need to avoid the super healthy participant that just want to be part of the preventive trial. When we designed the MAPT trial, near half of the population have got the high school diploma in France. At the same time, in the general population, only 17% of the people have got this diploma. So we have a huge selection of the population. So at that time, before this trial, because before we have another negative trial, but we say we need to understand why people accept to take part to preventive trial and why people refuse to take part to the preventive trial. And so we got the funding from the IRESP in order to analyze the determinants of the participation of the people when we ask them to enter, at the time when we ask them to enter in the preventive trial. And we use quantitative and qualitative approach. So we were lucky because usually the ethical committee don't like that we go to see the population who refuse to take part to the trial in order to understand why. But because it was maybe the main aim of our trial, our study, they accept that. So we have more than 50% of the people who refuse to take part of the large preventive trial that accept to be followed by our survey and accept to be even interviewed by a sociologist and a psychologist. So what is the main results of that study? Of course, we observed expected determinants of participation. The people with a high level of education participate more. The people who perceive the high level of risk to develop Alzheimer's disease was more likely to participate. But we have also some surprise. The main reason to refuse, the people say, I have no time, your trial is too demanding, there is a lot of burden, I have no time, no way to come 3 times a week for 1 month, 2 times a week for another month. It's just completely impossible for me. But when the sociologists go to see them in an interview during 1 or 2 hours, they got 2 information. The first one is that the concept of prevention was not completely understood by the elderly people. They say, I have nothing. My first baseline analysis is normal. Why I need to do something? Because I'm not sick. The second point is the credibility of the intervention on cognition. They didn't trust the link between nutrition and cognition, between physical activity and nutrition. So we need to better explain in general message and mass media message about that. And the third point was the fact that, at least in France, the representation of the disease in the media is a catastrophe. So in fact, the people say, I have nothing to do. Alzheimer's disease is a terrible disease. I have nothing to do. The second challenge is the amount of intervention. When we are doing a clinical trial with a drug, before we are naive to the drug. When we are doing a multi-domain intervention, we are not naive of diet, we are not naive of physical activity, so we need to take that into account, and it's not very easy with the tools that we have. We need also to pay attention about the adherence during the study period, but also the maintenance of the behavior change after the end of the trial, because sometimes we have a lag effect, so it takes a long time to observe an effect on cognition. What about this adherence? We were able to mix our data with a finger trial, and we saw that the adherence decreased dramatically with the complexity of the intervention and with the intensity of the intervention. And for example, in finger, it was very easy for the population to accept the visit of the nurse at home and to have this assessment of cardiovascular risk factors. But at the same time, less than 30% accept the cognitive intervention. In MAPT, it seemed that adherence was okay, but if we are looking the very end period of the trial, only 17% of the population in the intervention group continue to do what they are supposed to do. So after 3 years, it's almost impossible to maintain the behavior change that we can expect at the beginning of the trial. And this also was found in the live trial, the publication of Marco Power, that found that when you ask people to change their habits in the field of physical activity, It's okay for the 6 first months, okay for 1 year, okay for 2 years, but at 3 years, all decrease. And at the same time, the control groups start to increase a little bit their physical activity, so you can find nothing. So just to say that when we are doing a clinical trial, we don't expect to replicate what's happened in the real life according to the life course epidemiology. We know that when we are doing a trial, we are doing something at one point, which sometimes is not a good point, with a single exposure, with a non-cumulative effect. We are just unable to assess that. We have just one specific window of exposure. It's not maybe the good one. So this morning, somebody saying, we don't need trial. I think we need trials. I think we need trial because until now it's the best level of evidence. And really, I think if we want to change the habits of the population, we need to convince them with some data. We need to assess also the effect size, the benefit that we can observe, and at the population level, we need to assess the cost effectiveness of what we are doing. So to conclude, I will speak about the perspective of the future trial. And like it was done yesterday and said, I think we need larger and larger trial and long-term trial because we are addressing a population elderly. I think elderly is more than 50. So we need to address the complexity of this population because they have lots of comorbidity. It is not the way to say, oh, they have lots of comorbidity, it's too complicated. I say that because when we ask for funding, sometimes people say aging is too complex. So no way you can go to another way to obtain the funding. We need to adjust the aim of the trial because all these trials that I present say that it's prevention of multi-domain prevention, but it could be very different if you want to enhance cognitive reserve in very normal population, if you want to maintain brain health in the population who start to decline, or if you want just to prevent AD in people who are really at risk. Another point is to better target the population in the future because the participants included in all that trial were selected based on the calendar age and selected on the normal cognitive function that was evaluated at baseline. And in fact, we know that already 30% of this population have already amyloid in the brain. So maybe in the near future, the blood biomarkers will change the field because it will be easy to implement that in the large sample in this kind of trial. Maybe the biological age will help us a lot, also more than just the calendar age, which means nothing for me. To conclude, I will say that we still need some kind of good surrogate because we need long-term study. We can't do trial for 20 years, so we need to have very good surrogate like it was done in the field of cardiovascular. And they succeed, but the large trial were quite recent, I think. There is a very good trials recent. So if we need to, if we succeed, the problem will still big because we will need to implement what we observed in trial in the general population. And this, I think, is more than science. We need to have the politicians with them. And in some countries, it's very difficult. So this is my last slide. Yesterday, we spoke about the funding. 5 years is not enough. 10 years is maybe not enough because between the time when we think about a clinical trial and the time when you can publish, even if it is negative, it takes longer time. So you spend 15 years of your life. Thank you.
Thank you, Sandrine. It was super important to emphasize the importance of the trials in that field, which is so complex. I have 2 questions for you. I would be interested to get your experience on the building of those trials and explain us how you choose the risk factors on which you want to intervene with these multi-domain trials, because it seems that there is a lot of heterogeneity across trials. And for people like me who are not in the trial field, it looks like a little bit people are doing a bet on a combination, probably based on very logical, based on what they see in the clinics. But I'm wondering if we could maybe make more intelligent use of observational data to simulate the optimal combinations for future trials, and maybe also figure out on how to incorporate global exposures which do not rely only on individual choices and behaviors, but how can we foresee to integrate, for instance, air pollution exposures and things like that. So very complex question, but what is in practice? How do people think about this when they build the trials?
Yeah, in practice, it's very pragmatic. So in fact, when we are doing a large trial, we know that we have an amount of money. We know that we have the sample size is calculated in advance in order to be effective. And after that, we need to see your more you have some risk factors, your more you are likely to see an event during the period of the trial. But the more you have some, I will say, early signs of something, maybe it's too late. So it's always a combination about that. When we did the GECC trial years ago, we decided to include the population who go to see their GP because they have subjective memory complaint. And we say, if something works, in this population, that will be finally the population that could benefit from something. Well, we tested at that time antioxidant. But in fact, by doing that, it was very pragmatic. Now, I think I will just go through the risk factors for Alzheimer's disease, but not already subjective memory complaint, not already amnestic MCI. For me, it's maybe a little bit late to do that. For the second question, is very challenging. And in fact, the reason why I speak about biological age is because if in our trial we have biobanks that we can follow every year, we can have biomarkers of many things, but it's difficult sometimes to have the big range of the exposition because the number of sites are limited. And it's voluntary to be limited because if not, you increase, I will say, the heterogeneous measurement of cognition. So we wanted the quality of the clinical trial in this preventive trial. So we reduce the number of sites, but by doing that, we reduce the range of exposition in many things. But I think there is something more to do that we were doing until now. Yeah.
Thanks a lot for your presentation. I have two questions. One is, I'm not sure I fully agree with you that when you say that you need to convince the politician, the politician or whatever, the deciders, because if you think about the pointer trial, it's not sustainable to be applied to the whole population. So isn't it the other way around, that the trial should be thought in advance with the decision maker to make sure that it's implementable at a large scale? Otherwise, we are pleased between researchers, but I'm not sure it's going to be implementable, and it's a big problem. And the other question is, there are some criticisms about pointer, saying that it's rather a practice effect or whatever. So what do you think of that?
Yeah. So for the first question about the fact that we need to have politicians with us, I think that at least in France, maybe in US also, we need to give science for politicians in order to give us the best data in order to take clear decision. And I think that we are in our own world, just scientists doing good papers, and everybody in the room knows the paper of the other one, but except that room, it's difficult to go out of this world. And I say that because, of course, in pointer, it was maybe not sustainable, but in fact, she gave money. She gave money to the people who were adherent, and also she reimbursed the fact that the people go to eat some fresh fruit and vegetable. It was blueberries, but anyway, something that we need to think if it is. The politicians can decide that it will be easy, cheap to buy healthy diet than to buy bad diet. until now, if I'm going everywhere, it's very easy to buy junk diet, but very difficult to find good one. And if I find good one, it's very expensive. So I think this is is something that is politics. It's not scientific. Everybody knows that. Everybody knows that healthy diet is better than the junk diet, but the people who eat not so well, sometimes they have just no choice. That's the first question. And the second one, because now I have my CAGE score is increasing.
It was about the practice effect.
Yes, but yes, I did a comment about this paper about the practice effect. I think yes, but it's happened to many of these type of trial. Yeah.
Hello? Okay. I agree with you about the necessity of surrogates, but I agree more with the sentiment that came from the previous question asker. So if we assume that we can trust just the positive results and disregard the null, if we assume that the small effects that we see are truly meaningful to people, if we assume that all that we're throwing at people, do more physical exercise, eat better, do this and that, change your entire life, is sustainable, is it really the right solution to do more trials with a longer time frame if we might just end up with 2 intervention groups that are pretty much the same and a huge percentage that have just been lost to attrition?
Yeah, I think yes, because what I saw when we are analyzing deeply all this trial, there is a huge variability of the population, of the duration. Some of the trial are 3 months of intervention, some are 6 years. There is nothing to compare, in fact. So if we have one day a big trial that help us to analyze subgroup and maybe to decide one outcome that have value for everybody, it will be okay. The clinical value of the change in 1 point or 2 points on a cognitive scale, I agree with you, is something very difficult to admit from the population. They don't have to expect something that is visible. In fact, when we are doing prevention, it's like when you brush your teeth every morning, you can see nothing, but you do that. And I think the solution is maybe to learn to the young people to have more healthy diet, healthy way to do physical activity, to avoid us. Because why we are doing clinical trials? Because some people at one point have not chosen or not, they don't have the good physical activity level and they don't have the good diet. So our aim is to see, is it possible to change the outcome in that population if we change the habits, or if it is too late, if it's not enough, etc., etc. So I think it's important to demonstrate that. And maybe for another point, I would say that I'm not somebody who is fond of physical activity. My father will kill me. He was a rugbyman professional. But I need to have proven data that maybe will change something for my life if I have to do physical activity.